1950s. And they made a very, very important discovery. His name was Harold von Brunhut. And I know you're all sitting there thinking, I should know who this is. But what he did was, and as every comic book reader knew, and every tabloid reader knew, for the mere price of 75 cents, or $1.25 in my day, you could send your pre-addressed stamped envelope off to a clearinghouse in New York, and you'd get back a bag of sand. And in that bag of sand contained instant life. Sea monkeys. You can still buy sea monkeys, but sea monkeys are actually a real critter. They're a crustacean, just like a lobster. And they go through this amazing life cycle. So what happens to them is, even though they live in water, they can lay their eggs, and those eggs can become completely dehydrated. And then all you have to do is add water. 1,000 years later, long enough for them to hang out in a clearinghouse so Harold von Brunhut could sell them, and you can buy them, and they come to life. So you can still buy these things. They're about £2.50 on Amazon. So you have to buy about 10 of them in order to get your discount for delivery. Or you can, if you're kind of worried about the dubious claims that they're going to build little sand castles, and they might do that in your sink, you can go slightly more upscale, pretend like you're a naturalist, and buy Try-Up World, which is about £7. And you get free delivery on that, I think, because they're trying to push it. So I did this. I bought some Try-Ups, and I tried to time stamp the day that I did it with some news. But unfortunately, the news is going back and forth on this issue a lot, so it doesn't really work. But anyway, here's my Try-Ups in my dark kitchen in Oxford, and I grew some sea monkeys. And this is what they look like about 5 months later. Now, amazingly, they swim around. Now, the keen eyes among you will realize that this is a different container, and it's because I ripped this off of YouTube because mine all died, okay? And this is why I'm a physical scientist and not a naturalist. Okay, but the interesting thing about this is you just add water and you get life, okay? Now, you can't just add any old liquid, so if you think about some other liquids in your life, octane's a good one. So octane's what you run your car on, and if you're like me and you really like fast 1960s cars before we knew they weren't environmentally friendly, you want to run your car on high octane. It's part of my birthright. I'm supposed to like it, okay? But if you put sea monkeys in octane, they will not live, okay? They will die, and I didn't do that in the interest of not harming any sea monkeys in the making of this lecture, okay? But this is what they look like. So if you look at these two liquids, this is what they look like. This is hexane, and this is water, okay? And what do they look like? Clear liquids, really exciting. They kind of move the same, right? They're both in the same container. This one looks a little bit different. If I shake it up, I get bubbles. They look the same. So why can I not do this? Well, it turns out, now this is hexane. This is not octane because I don't have any octane. This was in my lab, and so I took the convenient thing in my lab. But hexane is actually quite similar to octane. But it turns out, in order to really understand how this works, you actually have to look at water on the microscopic level. You can't just look at these bulk properties of a liquid and decide what it's going to do. So there's a difference between these two, and I'm going to draw for you, and hopefully this works. And this is what the molecular structure of water looks like. Okay? It's just an oxygen atom attached to two hydrogen atoms. And I'm sure you guys have seen this before. Maybe sometimes it's on the back of football jerseys these days, because chemistry is cool. And this is what hexane looks like. It's just a bunch of CH2 atoms connected together. It's a bit dull compared to water. Okay, but these both have hydrogens. So the keen eye among you will notice that one has an oxygen and one doesn't. That's the answer. You must say, okay, water has oxygen. Well, the thing about, and you would be right, in part, okay, because the thing about oxygen is all atoms have a positively charged center with electrons that rotate around them, okay? And oxygen really has a high propensity to pull negative things towards it, a bit like my grandmother. All negativity pulls towards the oxygen, okay? And it's a big oxygen hog. Okay, whereas hexane and carbon's perfectly happy to share, and so you actually end up with an even charge distribution across this molecule, which does not exist in water. So this is slightly negative charge. We do this little cool delta because we're scientists, but this shows that it's a slight charge and not a full charge. Okay, and then we have slightly positive charge here. And that's a tiny little bit of difference. So now some of you that have had a little bit of chemistry will be thinking, yeah, but come on, water is not the only liquid with oxygen in it. And you'd be right. Ethanol, everybody's favorite liquid, right? Everybody's favorite liquid next to water. And this is ethanol. And you can see that ethanol and water, hexane, ethanol and water, they look exactly the same, right? Now, if you put the sea monkeys in ethanol, you don't just get drunk sea monkeys, okay? They don't live, okay? And we have special molecules in our body that I'm going to talk more about that allow you to metabolize ethanol. But you also should never drink pure ethanol, okay? So this is what ethanol looks like. This is the molecular structure of ethanol, and it also has an oxygen in it. So what are we going to do now? And it also, oxygen has a high propensity to attract negative things towards it, and so it still has a slight negative charge. And hydrogen still has a slight positive charge, but the difference is the direction, okay? And this is a tiny little difference. This is one of these little differences that you think, yeah, it doesn't really matter, but it matters very, very, very much. Because in this, you get life, and in that, you don't. There's a huge difference. Okay, but water in itself is not sentient, okay? And you all know this because you don't have to worry that when you leave your tap on, that I seem to have lost, oh, there it is. When you leave your tap on, that water is going to, make some sort of, invite its friends over. Well, you might worry about this if you're a homeopath, but water's not going to invite its friends over. Play poker games in your kitchen. Water has not created the work of Shakespeare. It's just not going to happen. There's a lot of other molecules that have to do this. And what's everybody's favorite biological molecule? What molecule do we always love to talk about with life? DNA. Well, DNA is like the middle management of molecules. All it does is give something else the plan, right? So all it does is tell your body how to make proteins. Proteins do everything. So you're breathing right now, I hope. I hope you haven't passed out yet, right? And when you breathe, you have little proteins that carry the oxygen to all the muscles in your body. Okay, I'm walking around right now and I'm using my muscles to do that. And perhaps you're twitching to try to keep yourself awake, right? And you're using muscles to do that. And all that work happens with proteins, and DNA tells you how to make proteins. And that's it. So it's off at the bar having a metaphorical drink while proteins are doing all the work. Proteins are also the most diverse biological molecule we have, right? So they estimate, now nobody really knows what this is, but they estimate there's about 80 million proteins in the world, and that's a lot of stuff. But they're made from a surprisingly small number of molecules. And here they all are. So all of the proteins that you have in your body, your bones, your lungs, your muscles, your hemoglobin that carries oxygen to you, right? It's all made from these 20 molecules, okay? And that's it, which is kind of sad, isn't it? Okay, but how do we get this huge diversity of life from this tiny number of different molecules? How do we actually get that? So if we look here, right, they all have this same functional group on them. You can see them all here. Everybody's got one, right? So this is actually how your body joins these molecules together, so they're irrelevant. All that makes the difference here is these little bits on the end. So these are all different molecular structures that make a difference. Okay, now you all have a pipe cleaner, and you're probably wondering why you have a pipe cleaner. So what I want you to do while I'm talking, while of course paying most of your attention to me, is sort of play with your pipe cleaner and start folding them into something, okay? And don't look at your neighbors. In my group, don't cheat, okay? So don't look at your neighbor, right? Just kind of do it here on the sly, and we'll come back to it later. And you don't have to if you don't want to. You can just be left out. That's okay. So all of these things have to link together to make a protein, okay? And the way that these are amino acids here, and they all link together through these chemical groups to make something that looks like this, okay? And all they are is long chains of amino acids, one after the other, after the other, after the other, after the other, into these long polymer chains, okay? Now, and there's only 20 of these things, so how on earth do we get this amazing diversity of life from just 20 things? So most proteins are somewhere between 100 of these long to 50,000 of these things long to even 100,000. Okay, there's a huge number of different sized proteins and different shaped proteins, but different length proteins. So if we just imagine a minute one of our smaller proteins, which is only 100 amino acid long, it's pretty tiny, okay? And we think about how we can combine that. So we have a 100 amino acid protein. You have 20 options at each position. One position doesn't presuppose another position. It's completely random. You end up with that number of different options, okay? And that number turns out to be, does anybody know? Off the top of your head? Many, many zeros? That is 10 duodecillion. I didn't know that number until this week. I didn't know a number that big existed, okay? But that is 10 duodecillion. That's a whole lot of diversity. So if you all stop for a minute and look at your proteins, and look at your thing. Does anybody have ones that, without cheating, that look the same as their neighbors? So you know what? This is why we need DNA. And I reluctantly say this is why we need DNA, because we don't end up with this. Okay, we end up with something that's directed. We end up with something that gives us the right structure, and that's all DNA really does for us. Okay, occasionally it makes a virus, occasionally it cures cancer, whatever. Okay, so this is what proteins really look like. We're not just big, long, spliff-like creatures with long polymer chains of proteins. They actually have to fold up into structures to be something. So this one here is hemoglobin, okay? So this is how you're breathing right now, and it's happening really fast. Okay, if you get too much acid in your blood, so if you ever sprint too fast, like for the train, your muscles will hurt. It's because you build up something called lactic acid. You have a protein to sort that out for you, okay? This is a muscle fiber, and if you want to be really super cool, you can actually make videos of these. And all that happens when you contract your muscles is that you have proteins doing this. Okay, so that's a little pulley protein. It's called actin, and it's going to say motor protein here in a minute for me. Thank you. And this can walk along your muscle fibers, okay? And protein, do you see any DNA? I don't see any DNA. Proteins are doing all the work, yeah? And all this work has to take place in solution, okay? So we have proteins, and we have water. And we know that somehow, so I just told you that DNA helps us make these long chains of molecules, right? And here he is here, right? But it doesn't just have a long chain. It has to fold up into this structure. And this is the miracle. This is the really interesting part, right? You can take this protein, and you can put this protein into solution, and every time it will form that structure over and over and over and over again. And I don't mean sort of like that structure, like your lovely pipe cleaners. I mean that structure exactly. If it didn't, we wouldn't be here. We wouldn't be alive. Okay, and that's amazing. That's absolutely amazing that you have all these amino acids and just water and code to do that. And there's another famous man in the 1950s, maybe not quite so famous as Harold von Brunhut, right? And his name was Anfinsen. And what he did was this exactly. He took egg white lysozyme, so it's just the protein in a hen egg, right, which we eat all the time. And he took that protein and he put it in water and he looked at the structure of it. And then he put it in a chemical called urea that unfolds it. And then he took it out and it refolded. And he put it refolded. And he did this and he did this. It must have been great being one of his graduate students, right? Because that's what you did for months and months. And people have repeated this, so he's not the only person that's ever done this. So you can fold and unfold, and you can do that all day long, and it works. And the only thing that you have to do is have the right amino acid sequence and have water. Instant life, just add water. Now, if this doesn't work, the reason why we study this is because it's cool. It is really cool, right? Nobody knows how this works. But the other reason is that this is linked to lots and lots of diseases. So Alzheimer's linked to proteins not folding properly. So in Alzheimer's, what happens is the proteins unfold and then they start plaquing together. Sickle cell anemia, okay? Sickle cell anemia is a disease of hemoglobin. One amino acid changes and the whole thing falls apart. One amino acid, the whole thing falls apart. So these tiny little changes in electronics and molecular structures make the world of difference to whether or not you're alive, okay? So we have, of course, a theory about this because we're scientists and we like to have theories about things. And the theory goes as followed. It's something called the hydrophobic effect. All right, and now I'm going to do my one demonstration, which hopefully, so I have been supported by Amazon, but also by all of the grocery store chains around Britain. So I have some Sainsbury's salt, that's easy to buy, and I bought the cheap kind. And olive oil, okay? So some of you cook, I know, and when you put olive oil and salt, what's going to happen? Does anybody want to guess what's going to happen? So it just sits there on the bottom. It's really quite boring, right? So oil is hydrophobic, okay? Oil, and you know this. You know, you walk around when it's in puddles in London, and you see this oil sheen on the surface of those puddles. That's because the oil is hydrophobic and it floats on top. So if you put this in here, so the theory goes, salt is hydrophilic. So salt loves water, oil hates water. So what happens is this can't dissolve. So you have this thing that's hydrophilic that can't dissolve in something hydrophobic. And then all you've got to do is add water. Just for your information, the theory on this is very long technical papers. It's not just this experiment. So if you take this and you swirl it around, what you notice is that the salt's starting to dissolve, okay? But the salt's not dissolving in the oil, the salt's dissolving in the water because it's hydrophilic. And I might have to use something to stir this up, but we'll come back to this in a minute since it's a slow process, okay? And that's all it is. So the theory goes that you have this hydrophobic stuff, and it's afraid of water. So water actually inspires it somehow to fold into the inside, okay? So all the hydrophobic parts of the protein, because some of those amino acids are hydrophobic, some of them are hydrophilic, they actually fold in together and make your protein. And voila, magic, you have a protein. And hopefully that will actually dissolve. But I was told by the nice people that helped me arrange this that if my experiment didn't work, I could just say, well, this is real science, okay? So here it is, hydrophobic effect theory. So is there any evidence for this? Okay. So the theory gets a little bit more complex than just oil and water. And what we know from this theory, so we know from the second law of thermodynamics, which is the best scientific law we have, that any spontaneous change must increase the entropy of the universe. This is a law, okay? Proteins fold spontaneously. Therefore, the entropy of the universe must increase. Now, this is true, okay? And so the folding of proteins has been linked to a favorable entropic process. But what the theory tries to do, so this is a macroscopic observation. So it's just like me looking at these big flasks full of a liquid, okay? Entropy measures a big ensemble, which is this liquid. It doesn't look at things on the microscopic level, all right? And so the theory goes as follows. The theory goes is that I have this peptide, here he is, and water around these hydrophobic parts orders itself. So in liquid water, water's perfectly happy to dissolve the hydrophilic bits, which are the oxygen and hydrogen and nitrogen containing bits. But around these big chunky carbon bits, so with these lines, at the apex of all these lines is a carbon. Okay, so that's what those look like. And that water is structurally ordered around there. Water is not happy like that. Okay, and I'm sorry I said happy. I always get mad at my students when they say water is happy, because I always say that water actually doesn't have any emotions, and so don't say that. And now here I am saying it. But we're at a public lecture, so it's okay, right? So water is structurally organized. This lowers the entropy of water. And then water doesn't like that, so water goes flying off into the atmosphere or back with other water molecules where it's entropically more favorable, and that's where the magic happens. Now, is this a good theory? Well, it's a theory that is nice to explain. I'm hoping that you all understood it. But there's no evidence about this theory at all, not one shred, okay? So all we know is proteins spontaneously fold in solution, and that must increase the entropy of the universe. But we have no scientific evidence of this water structuring problem. Nobody has ever seen that. It's all been done by a computer model, and there's nothing wrong with the computer models, right? And this is a nice thing about scientific theories, is they work until they don't, okay? And when you have new pieces of evidence, or if you're a physicist, a new observable, right, then you have to change your theory, you have to adapt. And that's one of the things that we're trying to look at, is how water, what does water do? How does water actually interact with proteins in order to help them fold? Diffraction. So this is animation to try to lull you into submission, right? It's try to hypnotize you, so now you're going to believe everything that I say. So I'm telling you this other theory might be wrong, and now you can just stare at this, and then I can go off down to the pub, and you can just continue to stare. But what we use is something called diffraction, and there's a lot of wonderful RI talks like this one that are on the web that talk about diffraction. I think Jim Akalili's done one, so if you're really interested in diffraction, you can go watch one of those. But I'm just going to go through it quite briefly. So what you can see is there's two sort of slits here, right? This is just a double slit experiment, okay? But you can imagine those slits not being slits, but rather being atoms. So I have an atom here, and I have an atom here, okay? And what happens is I have this wave of something coming towards me, but because it's sort of a particle and sort of a wave, the particle can hit me, and then it spreads off as a wave. And what I get from this is I have a distance between these, and I get this characteristic wave pattern between the two, yeah? So what you do is you have a probe coming in, bounces off the two atoms, they both have two waves, and you can look at that characteristic wave pattern, and that can tell you where the positions of the atoms are. So it's a bit like being at a pond, a nice mill pond, and throwing in a pebble, and what happens when you throw in a pebble? You get ripples, right? And what happens when you throw in two pebbles simultaneously? You get ripples, and they either constructively or destructively interfere, if you want to use techie scientists talk, or you can see that the waves either get bigger or they cancel each other out. And that's all we're measuring is those characteristic wave patterns. And now most people's favorite diffraction probe is a X-ray. Everybody loves X-rays. It's like DNA. X-rays and DNA are our favorite molecules and favorite probes. But X-rays are really lousy to measure water, okay? So they're really bad at that because if you look, this is a relative intensity of scattering. So X-rays scatter off electrons. So all atoms have a nuclei, which is positively charged, which is relatively small. They're all orbited by electrons, which makes a relatively large cloud, okay? And X-rays scatter off of those. Now, if you're a uranium atom, that's really good because you're really visible. If you're a hydrogen atom, that's really bad because it's small and it's hard to see. Okay, so what do we use? We use something called neutron diffraction. So the picture on your left is, I think, yeah, your left is an X-ray image of a cigarette lighter. So it's just like when you go to the doctor. If you break your arm and you get an X-ray, when you put your arm on that imager, what you see is the bone and not the fat. Okay, so here you can see the metal and not the butane. But the one on the right is a neutron image of a cigarette lighter, and what you can see is the butane, which has a whole lot of hydrogen in it, and that's what you can see. So that's why we use neutrons. Now, you have to get neutrons from somewhere. You can either get them from a nuclear reactor, or you can get them from one of the best sources in the world. So you don't have to go to Paris, you don't have to go to Milan, you can go to Didcot. And Didcot actually is the home of one of the best neutron sources in the world, arguably the best. Of course, I think it's the best because I use it. And what it does is it creates neutrons through a particle accelerator. And here's another plug. If you want to learn about particle accelerators, Susie Sheehy does a really nice series for this institution on that. But this is basically how it works. This is just a brief overview of how it works. So basically, if you look at the red thing there, that's your particle accelerator. So you have a hydronium ion source. And all that is a hydrogen, has a proton and two electrons. All right? And it's producing those as we run down the red bit. So here it comes. There it goes. And so then they start going faster and faster and faster, and you can control them with magnets. Okay? It's got a charge. You can control it with a magnet. Then it goes into the synchrotron bit, which is this bit, this swirly yellow bit. And they start going about 97% the speed of light, and they go so fast that you can actually pull the electrons off them, and you're left with a naked proton, okay? Then you take that naked proton, and let's go around the circle again, and you bombard a target. So you're basically throwing it as hard as you can at a big neutron target. And in this case, I think it's tungsten, so it's tungsten metal. It could be uranium. You've all heard of uranium. And what happens is you have all these neutrons that go flying off in every direction. Now, neutrons are neutral. That's why they're called neutrons, right? And they're very hard to control. So what you have to do first is slow them down because they come off really fast, and you slow them down. You slow them down so you can get your matter, and you do that by running them through a very, very thick chemical where they have lots of collisions and they slow down in energy. So it's a bit like trying to herd cats, right? So protons are pretty, compliant, and they do what the magnet tells them, but these things come off in all different directions. But the nice thing is you can reflect them. So they go bouncing down the collimator, and then they finally get to your sample, and then we can finally do some diffraction, which is great. And those are created by ICES. So here we go, back to mesmerization, diffraction. We love diffraction. Diffraction, all you're measuring is the wave pattern when you scatter off of two atoms. That's it, but we're doing that with neutrons. Okay, so this is what they look like. We don't just measure the waves, we measure this wave pattern, and you can tell this sort of looks like the cross-section of some of these waves, I hope. So in this example, you're looking at the distance between two blue atoms, and this is real measured data for a different sample, but that's the distance between those atoms. Now, if you have blue atoms and red atoms, you're going to have blue-blue interactions, blue-red interactions, and red-red interactions. And then you can sum all those together and get the plot on the bottom. Okay, and that's exactly what we measure. But you can see instantly we have a problem. That's what we really have. And I'm also very pleased by this picture because I finally found a use for Damien Hirst. And here's all the different atoms that we have, okay? And we have to measure that, and all the distances within here, all at the same time. So all of our diffraction patterns carry all this information. So what are we going to do? What we're going to do is we're going to do something that's really cool. It's called neutron diffraction with isotopic substitution, which is a long word for just saying that deuterium and hydrogen scatter different. So differently, because I'm not still in Tennessee, right? So you have hydrogen nucleus. Hydrogen nucleus has one proton and one electron. A deuterium nucleus has a neutron, a proton, and an electron, okay? And interestingly, they have different scattering patterns. So I can turn that into that. So I can label everything so that I can see different bits in my samples. And to explain this to you, I'd like you to all stand up. It's a good time to stand up. Okay, so if I walk in this room and I look around at you, there is no way I'm going to remember where you're all sitting, right? A, because there's lights in my face, and B, because you're all different. But if I could have you sit down, I can measure a pattern, so I can measure a pattern with just you standing and just you standing, so I know you're there, and I know you're there, and you've gone away. Now, if I can have you sit down, I can measure another diffraction pattern that just has you standing up, and you're gone, and you're gone. And if you want to sit down, then I can measure this as well. So I have no information, right? So if I can just have all of you stand up again, then I can use an X-ray begrudgingly, okay? You are all deuterium, and I'm going to show you what that looks like just right now. And you can sit back down again if you like. So that's what it looks like, okay? So the cool thing about this is I can pick any sample I want to, and you know that I'm going to get to measuring some kind of, I promise I'll get there, measuring some sort of protein in water. And you can see that I have all these different isotopic substitution things. And this is all the same sample, okay? It's the same sample with different deuterium and hydrogen labels. And I hope you can tell that that's different, okay? So they all look slightly different. You guys are the second one down from the top. And once you start standing and sitting and standing and sitting, I can mute different parts of you out. And that allows me to combine my patterns in order to figure out where each atom is, okay? And now I have another problem. So this is nice, right? Does anybody know about crystallography? Yeah, some of you know about crystallography, great. Guess what? That's not a crystal. We don't have a crystal, we're in a liquid. We don't care about a crystal. In a crystal, it's like 4 degrees Kelvin if you want to look at a protein where water is not moving, right? We want to see the water, and we want to see proteins as they fold, not after they've folded. In reality, this is what we have. We have our molecules moving around, and we're getting characteristic wave patterns off of them, and they're moving, and we put deuterium on them, and that's fine, but maybe it goes away, and it comes back and goes away. So we have a really complicated problem. It's just a lot of atoms moving around. But we still measure characteristic wave patterns so that we get the average picture of what's going on, which is cool. Right, so this is real data. It wasn't drawn by Damien Hirst or any of my graduate students. It was actually measured. I've just put it in nice colors because it's easier to see. Now, the other problem we have is it's not quite that simple because we do what every scientist does nowadays, and that is we use a computer simulation. Because we have so many different interactions between all our molecules in here, we have to simulate them in a computer. But this is pretty easy, right? What we do is we get a box of molecules on your computer. It's kind of like playing a video game. At least this is what I try to convince my students. I don't think they believe me yet. But you take the molecules that you've just measured. You know what you've measured, you hope, right? Because you can measure it, you can weigh out a bit of peptide, you can weigh out a bit of water, and then you can actually put it in your neutron beam, and you can measure a pattern from it, and you know what it is. And so you build a box of molecules on your computer, and then you move around those molecules until you actually get them in the right structure that compares with your data. So it's called fitting your data. So this is one thing that we do that's really unique, and most people that use computers don't do this, is that we actually take our data and use that as a constraint for our computer model. And that's really cool because we know at the end we're going to have a model that's consistent with our measured data. Right, so finally, some of the stuff that we measure. So this is, again, real data. And if you can see, there's a black line, which you can't see very well in this graph, and there's all these colored dots. And all those colored dots are our measured data. The black line is our computer simulation of that data. Okay? Now, what we want to do, and the line in the middle is the difference between the two. So we measured this little tiny peptide to help solve our problem. And the reason why we measured this peptide is because it's known to occur in a lot of proteins that fold. So proteins fold, and they have to turn around over on themselves. As you know from your lovely playing with your pipe cleaners, is that you actually have to form some sort of turn to form a complex structure. And that's why we picked it, and we have this 1960s picture of it. So you have to turn them up. But what we actually measure in solution, I just said they were moving. So if you all can hold up your thing, so hold both ends of them. You can keep them folded. Some of you have a big blob, so maybe that won't work so well, but just pretend like you're holding each end, right? So what I'm actually measuring in solution is a snapshot of you all at the same time, right? So I'm measuring all of this all at the same time. I'm measuring them open, I'm measuring them closed, and I'm actually adding those together, right? Because I want to look at the peptide when it's completely open, I want to look at it when it's halfway open, and I want to look at it when it's closed, because I want to know what the water's doing at each of those steps, okay? All right, so this is what we get out of our computer models. So what you can see is this is a small peptide. It's just a simple peptide, and it occurs in turns in proteins. So proteins have to turn back upon themselves. And what we do in our computer simulation is we measure this average structure. So we measure all the water and where it's most likely to go. We measure this for a variety of these molecules. So the first thing we learned about this molecule is it is indeed folding. So we went from this, which is wide open, to that, which is folded. Okay, so I've gone from this to this, and I'm measuring both in solution. And what this picture is this is a three-dimensional representation of where the water goes. So oxygen atoms are always red, nitrogen atoms are always blue. I have no idea why. Hydrogen atoms are always white. So this is the hydration around one of the oxygen atoms, and it looks like a halo. It's a really nice halo, isn't it? And I hope you all can see that it looks like a halo. So what you can see, though, is that water is actually bonding to those. Remember that water, oxygen is really negative, so it really likes negativity. So what it's actually doing is drawing the waters to it. And it's put them on the sides of it because they start to repel each other. And that's really smart. But then we see this. We see something completely different. We see hydrogen exactly on top of this oxygen atom. It's just sitting on top there. And that's weird. That's not where it's supposed to be. So you're just going to have to trust me. This is what scientists do, isn't it? Like, trust me, I'm a scientist, right? Is that this is more normal, okay? This is a more normal thing. We usually see this hydration that's around the outside, and on the top, we see something different. So just to prove to you, I know this is a really scary slide, I know. But the reason why I'm showing this slide is because I don't want you to think that I just drew this on the back of an envelope on the way up here, okay? So you can draw these pretty pictures, but these pictures, a lot of maths goes into behind this picture, and I have a lot of really clever people that work for me that help me learn that we've learned how to do this. So basically all this flow chart shows is how we actually pick out what water's doing. So we can figure out where water is, and we can figure out where these patches of density are around our peptide, and that's really exciting. But what this graph shows you is that we can actually look at those patches of density here and here and here and here, and then we can map out the different angles to figure out exactly where a water molecule might go. So we know, remember, at the very beginning, I told you that we have this orientation of water, and it's only slightly different from what you have in ethanol, and that makes all the difference in the world. Okay? And here it is, making all the difference in the world, because there's only certain ways that water can orient itself. Okay? And that's really kind of cool. And we can actually map that out. So we take our neutron diffraction data, we take a box of molecules, we fit it to that data, we pull out the average hydration, and then we look at exactly what these water molecules are doing and what they're doing during each folding stage of the peptide. And then we end up with pictures like this. So all that work boils down to that, okay? But what you can see here is this is an oxygen atom, and this is a nitrogen. Hydrogen atom is the water is oriented perfectly to pull those ends together. So what we think water is doing is it's acting like the glue. So water's sticking itself to that oxygen, sticking itself to that nitrogen, and starting to get that protein to fold together. Now, of course, we're scientists, so the first thing we do is think, okay, that's probably wrong, so let's go test and make sure. We try to disprove our theory. This is what you should do if you're a good scientist. Sometimes that's kind of painful to do, right? You publish a paper, you're all excited, you have a new theory, and then you go and test it, and you're wrong. But it's part of the way we progress. So we're sort of 2 steps forward, 15 steps back. And scientists are people, so we do get stuck in our ways. So we measured this longer peptide, and one of my really excellent graduate students, she figured out how we could go through the simulation and actually categorize open versus medium versus closed, okay? And she spent a lot of time doing this, so it looks really good, but I hope you can see that you go from open to medium to closed. Open, medium, closed. And then what she did was she picked out the hydration patterns around different sites in the molecule, and this was the one that was different, okay? And it's right where we want it to be, whew, right? As you can see, this is a nice halo-y-like structure, so water's in a halo like it's supposed to be, and then as we close the peptide, the water gets clearly more oriented and localized in a certain place. All open and happy. As the peptide folds, it moves aside. Then we can also pull out this picture where we can find a sequence of events. So we start here with folding. We have a water here. The water reorients itself, and then we have closing. So we have water pulling, orienting, closing. Pulling, orienting, closing. Do you really want me to say that again, right? So I can keep saying that, but that's really neat. And it's only because of that tiny electrical charge in water that can happen. Right, so then we measured that, and we have 2 examples now, right? But 2 examples don't make the end of the story. So then we measured something else. We measured this really simple structure on the bottom. And again, this was all done with neutron diffraction. We also do other measurements as well. We do some NMR measurements, and we do different kinds of simulations, and we spend a lot of time trying to prove ourselves wrong. I mean, probably tediously so, but that's what our job is. And so what we noticed from this is a completely different molecule. Okay, this is a hydrophobic molecule. So remember before, we looked at, and look, all the salt's gone. Right, so all the salt's mostly gone. Okay, don't look at the bottom. Right, so all the salt's mostly gone because it's the hydrophobic bits and the hydrophilic bits. So we thought, well, you know what we're going to do? We're actually going to take a hydrophobic thing and we're going to stick it in water and see what happens. But we couldn't just stick it in water. We had to stick it in water and methanol mixture. And the reason why we had to do that is because it had to have a little hydrophobic surface or it won't go in, a bit like the oil. So what we thought was, well, okay, we've got this methanol molecule here. So this is the hydrophobic bit, and this is the hydrophilic bit, and this whole thing is meant to be hydrophobic. And we thought, well, all the hydrophobic things will stick together just like this. All the hydrophobic things in here are sticking together. But on a microscopic level, that is not true. So what you can see is this is methanol, so the OH bond is oriented right towards that hydrophobic thing. And now the water is oriented right towards that hydrophobic thing. That's wrong. It shouldn't be like that. And we want to know why, okay? But the interesting thing about that is it only happens in methanol. So we've measured, we don't just measure peptides. These are drugs, and I should probably give somebody a prize if they knew what drugs these are. So this is cocaine. So cocaine can easily cross into your brain. This is why people use cocaine. This is why cocaine is a very effective drug. Not just recreationally, people use it for medical purposes. Because one of the interesting properties about it is most drugs have to be both hydrophobic and hydrophilic in order to work. Okay, especially drugs that cross into your brain. So they have to go through your bloodstream, but they still have to be hydrophobic enough to get into your brain. And nobody really knows why. So we've measured a bunch of these as well, and we see the same sort of theme. Okay, these are all water molecules, they're different colors. So I have different students who have different color signatures, and I know who they are. But what happens is that we can see that water is actually mediating all of these structures. So the way the molecular structure seems to be happening or is controlled by water, and it's controlled specifically by an orientation of water. And we can only see this because we look at things on the microscopic level, and we do very, very, very careful measurements. Okay, so are we right? This is the hardest part about being a scientist. So we're looking at stuff that's right on the edge of what we can see. So all of this structure that we do, right? So what we do is we take all these measurements of microscopic structures. We spend lots of time seeing all these things. We try to look at water. We try to look at how things stick together, and are we correct? Or is this theory of hydrophobicity correct, where water is just really kind of uptight and disordered and doesn't move anywhere? Which one's correct? Well, we don't know. So the beauty about biochemistry is the following. So I'm a physical scientist, and I work in a biochemistry department, is that biochemistry has no periodic table. It doesn't, right? Chemistry has a periodic table. There are rules. There are things that your elements must do. If you put things together, you know how many protons you have, you know how many electrons you have, but biochemistry doesn't have that. So one of the really difficult things about this hydrophobic effect theory is because it's very hard to energetically deal with. And what I mean by that is the following, is that your unfolded protein and your folded protein have a very tiny energy difference. So normally what happens in a chemical reaction in a flask is you put two things together, and the thing with the lowest energy will form, right? And it will form spontaneously, and it'll be pretty obvious. But that doesn't happen in biochemistry. That doesn't happen in your body. Your body is a carefully controlled organism. So if your blood pH has to be about 7.4, that's it. And it can go to 7.2, and it can go down to 7.6, but it needs to maintain about 7.4. And the reason why it needs to do that is so that you can live. And your body can do that. And your proteins, if they're at 7.6, they unfold. If they're at 7, they unfold. They have to stay exactly at the right pH. So the energy landscape, to use a technical term, is very tiny. Okay, you don't have these big energetic changes and reactions like you do in chemistry. So what do you do about that? I don't know. So it's still in its discovery phase, right? So we're actually creating the periodic table of biochemistry. That's what we're doing. At least that's what we say that we're doing. Okay? So the thing that you need to ask yourself when you read science, so the thing about the thing about water and the way water orients is water is the only molecule that can do this. So there's been a lot of studies where people try to find water on the planets. It's never been, I don't think they've found it yet, because the way this happens is with life on Earth as well. Okay, so it's something to actually think about why we work. Now, the diseases related to protein folding are still not cured, okay? Most of the way that drugs work most of the way that drugs interact with your bodies are still unknown, okay? I don't know if you know this, but most drugs that you take, people don't know how they work, okay? Especially drugs that cross into your brain, which is one of the reasons why we're studying. They actually don't know the mechanisms of how these things work. There's not a lot of information about that. It's all sort of empirical. So part of the research we're doing is to try to uncover these mysteries and try to figure out why water is the elixir of life. And I'm actually done a little bit early, I think. So I think I can take some time for some questions.